Yeah.
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 Chalo, I think we can begin.
Can start.
Okay.
Vinayak, are we good to go?
Hey, good morning to all. On behalf of CNR Hall of Science and Education Technology Unit, I welcome you all for today's interactive lecture program in biology. We are having 45 minutes lecture followed by 15 minutes question and answer session. We urge you to actively participate in the question and answer session, which will be at the end of the presentation. You could ask your questions directly or comment your questions in the comment box. For YouTube users also, you can comment your questions in the comment box. Okay, during the session, I request you all of you mute your camera and microphone. Thank you all for your cooperation. Now, I request Professor Ravi Manjithaya sir to introduce our speaker.
Thank you, Vinayak, and good morning, everyone. My name is Ravi Manjithaya, and I'm an associate professor here in JNCASR, and I work on cell biology. And I'm told today that we have close to a dozen schools that are going to join for today's talk. These are 10th, 11th, and 12th standard students from Karnataka, Andhra, and all the way from Uttaranchal. So, we were just discussing with the speaker that life throws a lot of challenges, including this pandemic. Earlier, we could only accommodate so much in our classrooms, but now with the virtual classrooms, we get a bigger stage to, listen to talks. So with this, it gives me a great pleasure to introduce Professor Deepak Saini. Deepak Saini is an associate professor at Department of Molecular Reproduction Development and Genetics, Indian Institute of Science, one of the country's most premier institutes, research institutes in Bengaluru, here in Karnataka. He obtained his PhD from AIMS, New Delhi, and then he completed his postdoctoral research training in Washington's University School of Medicine, USA. And he's a faculty here in Indian Institute of Science since 2010. Professor Deepak Saini has pursued research in frontline areas of cell signaling, mainly age-associated inflammation, tissue injury, and infection. He looks at pathogenic bacterium, including Mycobacterium tuberculosis, which causes the TB. Okay, he's published more than 80 research papers, and I have close association with him for many years for his research. He's a prolific user of microscopy-based techniques. Okay, and it is therefore no wonder that his lab mantra is called "Seeing is Believing." And by today's lecture, you'll probably understand what he means by that mantra. He's very passionate about science communication, and he's always made himself available to reach out to school students despite his research-related responsibilities. And we are very grateful for your support, Deepak. And I'm sure all the students today will have a very eye-popping talk today. Okay, with this, I would like to invite Professor Saini to deliver his lecture to the student. One housekeeping announcement. I have been told by the organizers that in most of your schools, in your auditorium and such, they are projecting the talk as well. So in case you are in your school and instead of looking at your mobile, you want to see the things in big screen, which I strongly recommend because microscopy images look really good in projected projectors. So make use of that facility if it is available in your school. With this, back to you, Deepak. Thank you.
Thanks, Ravi, and thanks for the kind introduction. It's always a pleasure to come and give talk at JNC, especially talking to the young ones, because you never know amongst the audience, it might be a future student for all of us. So with that intent, I think I'm going to turn off my video so I can free up some bandwidth and you can actually see things clearly at your end. But feel free to let me know in case there is any issue. Is my video still visible?
Yeah, Deepak, your video is visible, but the way it has been arranged, it that now it's gone. Yeah.
Okay. So it takes a few seconds to turn off, right?
I think so. Yes.
Okay. There's some video also still on somewhere, but with one of the, presenters or yeah, great. It's still on something. Okay. Let's not worry about it.
Yeah.
So I think Ravi nicely introduced what he does and also what I do. I think at heart, most of us are biologists, and what we do is like to study biology using various screwdrivers which we have in our labs. So some of us use microscope as a screwdriver. When I say screwdriver, I basically mean tool. So a lot of time people give a lot of emphasis on tools, but what is fascinating about biology that the questions is what becomes what screwdriver should be used or what tool should be used. So we largely use microscopy as one of the tool to answer certain questions in biology. And I'll tell you why we use microscope, because there are certain things about microscopy which is far more fascinating than any other tools which is available to biologists. So Ravi, one thing I wanted to quickly ask, is questions allowed in between interruptions? If there's any, how will I get to know?
No, so what they will do is that there won't be an interruption during your presentation. They have two modes of asking questions. I think if I'm right, Vinayak, there is a chat box. The students are encouraged, and I'll take this opportunity to announce it to the teachers also, or whoever is facilitating these talks across all the schools, that you can somebody can collect these questions and type in the chat box. So at the end of the talk, it will be easy to moderate and ask those questions from the chat box, or we will also open it up for students to ask if they can address it. So it will be an uninterrupted talk, and then later we'll take question answers. So 45 minutes plus 15 minutes. That's the way we have it.
All
Yes,
right,
sir.
all right.
Yes, sir. Exactly.
Great. So this is the worst part of pandemic talks, that you're talking to the screen and you have no idea if the audience on the other side is sleeping, drinking a cup of tea, or actually playing video games, which seems to be the most popular hobby these days when you're listening or attending online classes, right? So anyways, I will presume that you guys are listening, and I'll also keep looking at my monitor, hoping it will respond to me. But anyway, let's keep those nitty-gritties aside, and let's talk about what I'm going to talk to you about. So roughly have 45 minutes to bore you guys. And if your phone battery dies in this 45 minutes, it's not my problem. But hopefully I will be able to keep you entertained for next 45 minutes. The idea here is to make you believe that microscopy can tell you some things differently. But to use microscope, I'm going to take a few steps back in history to explain why microscopy became such a powerful tool to look at biology. And also, a lot of times we wonder as students, at least I used to wonder, and I'm sure you guys wonder a little more or maybe a little less, all depends on how much of the Google has helped you answer your questions. But the kind of thing which used to, fascinate me, that what do we actually look under a microscope? What are we looking for? Are we looking for just something which looks very nice under the microscope, and then what? So, and if I say that I look on a microscope and do biology, the question you might want to ask is, what do we actually look under a microscope to answer in biological systems? So I'll take you through that. And so there are multiple model systems my group works on, and I think that is simply because um, we all have multiple interests. And second, we all also want to figure out various things because we do not know what would be the need of the hour in future in that particular area. As you would have noticed, nobody expected or
Hello,
wanted to
sir,
work.
excuse me, sir.
Yeah.
Sir, you're changing slides, sir. We cannot see.
No,
the
I'm
new
not
slide.
changing slide yet. I'm still in slide number 1. That's what I'm saying. I'm going to bore you guys for a pretty long time.
Okay, sir. Okay, thank you, sir.
It's okay, Deepak.
Yeah. So as you notice, what I was saying is nobody expected that somebody should be working on SARS-CoV-2 because that was not something of priority. But right now, it seems like half the world seems to be working on SARS-CoV-2 because it is a matter of priority. So a lot of us have various research interests because in future, we don't know which one will actually be area of interest. Now I've changed the slide. It's slide number 2 here. So this is a brief overview of what is this talk all about. And the first slide says, given that I'm a biologist, I'll talk about cells. But what I'm going to talk about is listed in these 5 points. How do we make sense in biology by looking into cells? What do we look for, look into cells if you want to try to understand some biological question? How do we generate different information in biology by using holistic? Holistic means full, intact methods compared to reductionist. And I'll explain for the students what do we mean by holistic and reductionist methods. Also, how do we redefine paradigm using microscopy? Paradigm is something which is written in textbook that this is what happens. And generally, if it is bold, it's more important. If it's bold and underlined, even more important is bold, underline, italics. It's a thing written in stone. But what I'm trying to tell you is there's nothing in biology which we can say it does not happen. Biology is constantly evolving and paradigms get redefined, rewritten by changing approaches, which is what we get flavor of in this presentation. I'll talk about microscopy as one of the tools. And lastly, I'm going to talk about how do we study transient physiological event. When you say transient, it means short-lived. Your ability to see by constantly moving your head or eyes allows you to see new things. It does not carry an impression of what you saw a few seconds back. That means the process of vision is very transient. It allows you to see, make an impression, record, and move on to take next image. things are very rapidly, is happening at a very rapid time scales. So how do we study such rapid transient events in biological system? And I'll tell you about a few things using microscopy. So in a nutshell, my talk is all about how problems in biology can be solved by using microscopy or imaging. And of course, when I talk about all these things, we have to show you that this can actually be solved by what I'm saying. So in a scientific terminology, what we say is we need to show you data. Data means this is what is happening, and we can actually show you inside the cell what I'm talking is indeed happening. So that is, in the end, I'll show you some movies from the cells. So sadly, the movies from the cells do not have an audio track because what cell talk we can't listen, and the way we talk we cannot comprehend. So it basically be silent movies from the black and white era, pretty much like black and white era. And I'll tell you what exactly those movies are telling us in terms of information. So my first question is, what is under the lens? This is something which piqued curiosity of a lot of rich people in the Renaissance. Renaissance, it's when a lot of people who were rich, had a lot of money to spend and burn, wanted to explore science, arts, literature, and even humanities in a different flavor by burning or spending their money. And this is a time when a lot of people in Italy, which is predominantly which is considered as the epicenter of scientific evolution, a lot of people were looking at things in a slightly different way. And this device, which is shown on the screen in the bottom right-hand side screen, is actually a microscope. And what is the best part of this microscope? It is made up of pure gold. Now, if you think about this, it's definitely not something which you would like to use for any scientific investigation or interrogation. It's actually a subject of or object of curiosity. It was a rich man's toy, which he showcased to his friends to show, look, I've got a device which can allow you to see things by making them look larger. The objective was very simple, to show people things which they cannot see with naked eye. And guess who made this first microscope? This first microscope was made by Leeuwenhoek. I mean, Leeuwenhoek is somebody all of us who study biology, who have studied science since school time, at least know about. Now, what was the profession of Leeuwenhoek? Leeuwenhoek was not a microscopist. Leeuwenhoek was not somebody who was interested in science per se. Leeuwenhoek was basically a merchant. What was his job? He was a merchant for the rich. And what we call now is curios seller. Curios sellers means he will sell objects of curiosity to anybody who's ready to pay the pretty money for that. So some rich guy wanted him to make a microscope or a lens or a magnifier so that he can see things which is not visible. And of course, Leeuwenhoek agreed to this demand and made a first microscope. It was slightly different looking, but he made a bunch of other microscope and lenses. He used to hand grind the lens and make such microscope or such devices. And Leeuwenhoek was also curious to know what he could see inside the things which was not visible by naked eye. And this is a very famous picture of Leeuwenhoek, which is coming from class 6 in textbooks. It's an image of a water droplet put under a hand microscope, which was made by Leeuwenhoek. And what does it show? It shows some moving animal cues, is what we call them, in the pond water. Left is amoeba, right is paramecium, and this is the earliest drawings which were there stored to show what Leeuwenhoek showed us. Now, what is fascinating about these pictures is that they show what we could not see with the naked eye. So what started as a curiosity became a major area of science when Robert Hooke started looking at various living things under much more magnified microscopes. It's something which allowed him to see things much better. The image in front of your screen is something which is notoriously popular. It is what we call as the image of the first cell because it looks like prison cells, and which is indeed true. And these are the famous cork cells which were imaged by Robert Hooke by taking a thin slice of cork wood and imaging under the microscope. Now, what is interesting about this? This image is by far the most famous image of Robert Hooke because presumably this is where the area of cell biology started. Because much later people realized what Hooke was seeing was actually cells, so they gave him credit of starting the area of cell biology. But Hooke was not looking for cell biology. Hooke was basically a person who was supremely curious, and he was also a very talented illustrationist. So he used to draw things, and he used to draw them so nicely that it actually, all the drawings which he drew for life he saw under the microscope as a book called Micrographia. For those who are interested in seeing this book is freely available on a website called Project Gutenberg, and you can download this book and look at what Robert Hooke drew. And for me, this particular image is one of the best images which I like from the book. This is actually head of a fly. Look at the detail. Look at the rendition which Robert Hooke put into the image of what is under a microscope. It's a fascinating view of something which you could not even appreciate by naked eye. So what started as a curiosity with Robert Hooke's and Lohnau's work slowly evolved into a major area of research. People actually started investing more resource, time, and efforts to investigate looking at things which was not visible by naked eye. So, but why should we be looking at what we cannot see with naked eye? The idea was we are looking at cells, and cells, according to textbooks, make up what we are. It's the cells which come together and make us a multicellular organism. But the point is, can we study cells to understand what happened to a multicellular organism like us, human beings, or any other eukaryotic system? Is looking at cell good enough? So let's look at some comparisons. Can we compare cells versus an organism to justify studying cells? I mean, basically, I'm saying, can we justify cell biology as a area of investigation or area one should study towards understanding more complicated organisms or a higher multicellular organism? So this is a very interesting picture of people walking. So one of the main features which we attribute to animal forms is they are mobile. Let's look at two small videos here. This is a video of amoeba. And what you see here is a single cellular organism which is being viewed under a microscope. And what you will notice is this amoeba is moving. And where is it moving? You can also see the nucleus, you can see the cytosol, and you can see some floating cytosol inside the cell in which some organelles are floating around. What is amoeba going to? Its amoeba was going to eat some food. It's engulfing food particles. So you can say, okay, this motion or movement can occur for organisms, but if they're single cellular, they do it by themselves. And if it's multicellular, you need a multicellular entity to walk around or to move. I'll show you another video. Now, this is a very interesting video. In this video, you can see certain round cells. These are basically red blood cells. And you can see a elongated cell in the middle. Is my cursor visible? The hand sign? Can anybody confirm, Ravi, if you're around, or Vinayak?
Yes, sir. Yes, sir. We can see, sir.
Great.
Clearly.
So this particular cell is a white blood cell. Basically, it's a neutrophil. And this thing right here is Staphylococcus aureus, a bacteria. And if you remember your high school biology or even middle school biology, white blood cells or neutrophils are the cells which will engulf the bacteria and destroy it. But the point is, how does a neutrophil know where the bacteria is? Does it keep on engulfing it and by chance it engulfs the bacteria also? Or there's some mechanism built into the process of sensing the bacteria and neutrophils, and it can actually attack the bacteria by the way it senses it and can reach it and destroy. So is there a way bacteria can be sensed by the neutrophil? So if you look at this video carefully, I'm going to play this video. And remember, this cell is from a multicellular organism, but this cell is in the fluid phase, which is the blood. And that means it's giving a medium in which it can move. But what I'm trying to show is that single cells in unicellular and multicellular organisms also are capable of moving. Neutrophil, Staph aureus, RBCs, and let's see what happens. Bacterium, and you can see that this cell is basically chasing the Staphylococcus aureus bacterium. And how is it chasing? It's pretty much chasing for the students who are there. It's like a dog chasing a cat or a cat chasing a mouse. It keeps chasing it till it finds a way to engulf and gobble it up. Now you can see it stops moving. It's achieved its job. The cell is actually moved around. It was actively able to sense the bacterium and engulf it. Now you might ask the question, this cell does not have eyes. How does it see where the bacteria is? It doesn't see by eyes, it sees by chemical cues. It's able to detect the chemicals which is secreted by the bacterium inside the blood, and it actually homes toward it. It's like, you're standing there and somebody's calling you. You keep hearing, going towards the sound because you can detect the intensity of the sound by if you're going closer to it. So such intensity-based targeting or motion, which we call as chemotaxis. Chemotaxis is moving towards chemical gradient, is what neutrophil uses to harm or home towards the bacteria. So what is the point I'm trying to make is, while we know the cells are building blocks and a large number of cells have to come together to make a multicellular organism like us, which is capable of a lot of very interesting behavior like walking, talking, eating, thinking, it is the fate of the cells in your body which decides what you will be. Cells per se are able to move, feed, communicate, reproduce, and die. And what is also more fascinating is that most diseases or disorders in our body are actually because of some things have gone wrong at a single cell level. Single cells can or actually serve as a good system to study physiological disorders. Cells, if a single cell goes wrong in your body and keeps proliferating, that is what leads to formation of cancer. It's a single cell in our body which gets invaded by a bacteria or a virus, which is not able to control that bacteria or virus, ends up causing systemic infection. It's a single cell in your brain or neuron which undergoes degradation or degeneration, which leads to neurodegeneration problem. It's things which are going wrong at single cell level, but ultimately it's a large number of cells which go bad, and that's why you end up getting a physiological disorder. Example is again diabetes, same thing happens in diabetes. So cells are regulating what is happening to us, and that makes cells a wonderful model systems to study. So cells are good model system for understanding basic biological function, and that's why we need to study cell biology. And that's a good enough reason for people in cell biology industry to keep on working and keep on trying to resolve things. Now let's look at cell. Now we have come to solving biological problems by looking at cells. So before we do that, let's look at simple, picture of cell, which is a classic textbook picture of cell. This occurs in various flavors in all the textbook. NCERT shows in different form, college textbook shows in different form, but more or less it's the same design which you will keep on seeing where the cell is shown. And what is the design? You see a round object and you see various intracellular organelles located inside the cell and which are aptly labeled by the artist who drew the picture. You see Golgi complex, lysosomes, flagella, plasma membrane, nucleus most prominently, endoplasmic reticulum. What you will find fascinating is if you look at a picture of cells between two artists' rendition, you'll realize that it has two interesting features, that not all the organelles are labeled by everybody the same way, one. Two, that the location of these organelles are not static. Every artist's renditions allows them the creative freedom to show these organelles at various locations inside the cell. But the question it makes you wonder that which rendition is correct, which image is right. Is it actually the artist's creative freedom, which is based on which he can or she can draw those organelles at various places, or indeed they are at different locations inside the cell? So the short answer is that these organelles indeed are very dynamic. They keep on moving around. They keep on talking to each other. So basically, when an artist draws a cell or you draw a cell, your teacher never cut marks because your nucleus is slightly less round than somebody else's nucleus, which is perfectly round. Nobody cuts a mark at nucleus on the left-hand side or versus the right-hand side because it is indeed true. Such type of freedom of location or size is there inside a cell. That means these organelles are very dynamic and they're in constant interaction with each other. They're talking to each other. Now, if the organelles could talk, what will they be talking? This is what they will be talking. There's a very interesting cartoon which I really like because it's drawn by a school student, and it actually very nicely captures what each organelle function is and what do they do. But the point is, how do we know that this is what each organelle does? That means we need some way to study these organelles and understand what these organelles are doing, and to understand at a molecular level, at a single molecule, at a single protein level, what these organelles, what makes these organelles, and how we can change their structure, function, and behavior. Because that's where most disease or pathologies emerge from. So what I'm asking you to do is, I'm asking you to look at a cell instead of an organism, because whatever happens at organism level can be seen at a cellular level. But what I'm also asking you to say, inside the cell, you got to study various organelles, because if something goes wrong, it generally goes wrong with one of the organelles, and that's what you need to study. Now, what you realize, I've actually made the microscopy problem far more complicated. I've asked you to not only look at cell, I want you to look inside the cell and see what's happening. Now, don't be alarmed yet, because I'm going to make it even more complicated in the next few slides. So we are trying to look at things. And this left image on the left shows you what we can see with various approaches. While our eyes allow us to see things which are visible to naked eye, which is all the way to the millimeter scale right here, anything below than that, we need to look at a light microscope, which allows us to appreciate cells and small tissue architecture, cell arrangement, and also probably inside the cell. And this is the visual spectrum. This is a wavelength which we can see. Remember, wavelength seeing does not necessarily mean the resolution. Basically, what we're seeing is a color of light. But anything below those wavelength, we are dealing with small molecules like DNA, RNA, proteins. And to see them, we cannot work with light microscope. We need something like electron microscope or atomic force microscope. Those techniques of zooming up the architecture generally takes image at very, very high resolution to allow us to appreciate the subtle differences in various cell types, organelles, and protein behavior inside the cell. Now, what is interesting here is the following. When you look at the scale of sizes, what can we translate into in terms of biological things? Things which are at a meter scale is where organisms, and in centimeter scale where we have organs and tissues. If we go below millimeter scale, we have cells. And if you go below micron level, which is where we have organelles, and what is what I want you to study. And also we have what we call as protein machines. Anything below a few hundred nanometers is where most molecules stay. Now this makes it a very interesting problem. That to study organism, we need to study organs, tissues, and for that cells. And to understand what is going on in cell, we need to study organelles or protein machine to understand what is happening to an organism at a multicellular scale. So this is a scaling problem. Now, what I'm ultimately telling you is to understand biology, which is even applicable to multicellular entity, we need to study proteins. Why? Proteins are the building blocks. Proteins are the one which make organelles. Organelles are the one which make cell. Cells are the one which makes organs and tissues, which make a multicellular organism. So now what we have done, we have broken down the problem to the basic building blocks, because it's easy to revert back and find something gone wrong at the building block to fix problems which has occurred at a larger scale. But the problem is far more complicated. It's easy to break the things down to the building block, but it's not easy to study the building block. How do you study proteins? Now, this is what we need to study proteins for. Because proteins, even though I say they are building block, proteins do far more things in our body. Proteins are not just structural entities of our body, they are also executors. They are enzymes in our body, metabolic enzymes, glycolysis, Krebs cycle, transcription, translation, everything is done by proteins. Proteins are pretty much doing everything which we know in our body of. Rhodopsin, the pigment in our eye which allows us to see, is a protein. Tastens, the receptors in our taste buds which allow us to taste, are proteins. Receptors which allow us to sense temperature, touch, all our senses are basically dictated by proteins in our body. So proteins are basically executors. Proteins are also regulators. What do you mean by regulator? Changes which occur in our body in response to a cue. If I throw a ball at you, you will duck. Proteins are regulating the response which is generated by the rhodopsin in the eye where they see the ball coming. But in the very short time span, which is few hundreds of milliseconds, less than a millisecond, your body has executed a series of events which allows you to duck the ball. So proteins are also regulatory. When you grow from childhood to an adult, your body undergoes a lot of changes, physiological changes, biological changes. Those are changes are regulated by transcription factors or regulators. Those are also proteins. Proteins are also communicators. As I said, they pass information from one part of the body to another. Hormones. Proteins are also fate deciders. When cells in your body decide not to respond to a protein, not to detect a protein, they undergo degeneration, they die. When proteins respond to the growth factor, which is also a protein, your body undergoes growth. You actually grow up. Proteins are all the senses. Proteins are also protectors, antibodies, which is what become very popular in this COVID age. Everything is housed at protein level. So what we got to do is we got to study proteins. So you see, this is all the protein molecules. They need to connect and light up the lamp. And that's the idea. I really like these guys because they'll actually help you understand far complicated concepts in a much simpler terms. So how do biologists like to study proteins? How many of you have fallen sick? Almost everybody, I presume. And in this COVID era, not falling sick is like a luxury. But when you go to a clinician, a doctor, the doctor asks you, go and get a few tests done. What are those tests when clinicians is asking you to do? Clinician is asking you to do not just record if there are the change in the cell number in your body, which is what they like to do. They're also looking for changes in the levels of certain proteins. Remember, they're saying, do you have inflammation? They're actually looking at the level of certain proteins in your body, like cytokines. They're also looking for activity changes in certain proteins. They say, is your liver enzyme, liver function, kidney function test good or bad? They're basically testing the activity of certain enzymes. They're also looking for changes in protein function or state. Proteins can be cleaved, chopped off. Protein can be modified. Protein undergo chemical changes. They can undergo oxidation. They can undergo attack by certain molecules like sulfur. They undergo sulfidation. They can undergo nitrosylation. So protein actually undergoes change in their chemistries. Then proteins inside the cell also change location. What do I mean by that? Let's say there is a protein which is living in the cell cytosol. This protein is able to tell the cells in your body, let's assume them to be blood cells, that in response to a pathogen, this protein should go into the nucleus and tell cells to divide. That means the DNA in the cell should undergo duplication to promote cell division. So basically what we're doing is we're trying, the proteins are also changing location. They're basically carrying the information from one part of the cell to another, one part of the body to another, and that's also we can study. Protein also, we can study protein by looking at metabolic molecules they generate. Let's say enzyme in glycolysis, which catalyzes changes in, say, glucose to glucose 6-phosphate, a classic enzyme which is called hexokinase. Now, I may not be able to measure hexokinase activity, but I can look at the levels of glucose 6-phosphate, which is an indication of hexokinase activity. So biologists study proteins either directly by looking at their levels, looking at their activity, looking at their modification, looking at the location, or looking at the changes in the molecule they catalyze the reaction for. So biologists love to study these things. But the point is, all these measurements tend to be static. You are sick on day 1, they look at the liver function. And day 2, they look at your liver function again, and they decide whether you're getting better or worse. They are not able to record it real time by any means, which will allow them to study if your condition is getting better or deteriorating. So what is required is that we need to study protein changes in a dynamic way, but what we end up doing is in a static way. And a lot of time, these measurements actually give you partial information, but what we like to collect is complete information. So there are a lot of problems with existing methods which biologists use to study proteins. They give you exaggerated readout. If you take a pure protein in a test tube and start studying it, you realize it is doing what it's supposed to do, but in a much different way than it might be working in the body. That means there is an overinterpretation of the protein behavior. Many times we do not know what a protein needs to show some activity. We keep taking the pure protein, trying to study, and we don't understand why we are not seeing what we're supposed to see. Protein also gives undesirable readout. If the protein is supposedly toxic, protein which triggers the cell death, we want to study it. We take the protein cell, put it to cell dies. There's nothing to study. The problem is whenever we're trying to take pure proteins trying to study, it becomes problematic. And also, looking at one protein at a time can give you limited information. And why I like this point is very clear. If you want to study proteins, you can imagine a cell is like a classroom with hundreds of students there. Each one of the students is actually one protein, which is doing some particular job. They can either be working in concert towards a particular objective, or they might be working independently. But if you actually think from this perspective, cell is like a stadium. In a stadium, there are hundreds and thousands of spectators, and each one of them is a protein molecule. Some of the spectators might be coming from same family, or same school, or a same geographical location. That particular group of protein might be doing the same function. And that could be a bunch of proteins which are part of a particular organelle. There are some proteins which will be very dynamic, like children. They're moving around and doing things inside the cell. And that's why studying protein becomes critical, but that also makes it very, very challenging. So we are talking about not just looking at a cell, but we're trying to ask you to look inside the cell. And if you keep taking these individually outside the stadium and trying to study them in isolation, you'll actually learn nothing. You take a person outside the stadium who's shouting and yelling and put them in a room and start studying them, you'll realize it does nothing. So what you end up getting is limited information and incomplete picture. Like the kid right here who was asked to draw what's under the table, what's under the microscope, he ended up drawing a table, which is indeed right, but it's also a wrong information. And lastly, those who have done biology, especially enzyme assays, you realize no matter how many times you do a biological experiment, the change always happens during the time in which when you have not done the experiment. That means when you do an experiment, you collect information at hour 1, 2, 3, 4, 5, because that's your time point. In between them, when you have not taken the sample, that's when the changes happen. Biologist is always running because there's always the next experiment time point. And that can be solved by looking things in the living cell. I'm going to show you some images. Can you tell me what this game is? Because you cannot talk, I presume all of you have actually seen that this game is football. When you look at these images, what do you see? You see that it looks like a game which is played by using a ball, and we kick a ball and presumably with the feet. I'll show you two more images. Now, if you look at these images, what you realize is now suddenly this game looks different because there are two individuals who are trying to touch the ball with their hand. So now you're confused if this ball game is played with the feet or hand. So the point is, images tell partial story. If I keep showing you pictures after pictures, you will learn very little. Sometimes you can learn, but it's always a lot of confusion, a lot of doubt, which is there. But if I show you a video of soccer for a few minutes, and let's say I show a video of a few minutes of soccer game, you will be able to understand soccer much better by looking at a video than looking at the pictures. And that is something which we need to keep in mind, that videos give you far better information. Now, what I'm telling you, that instead of looking at proteins by purifying them, by breaking apart, and this is what we call as the reductionist approach. Reductionist approach means break open a cell, purify proteins, and look at them in isolation. This is reductionist, breaking the problem down to individual bits and pieces and studying them. We want you to study things inside the cell without breaking open the cell. Now, that means we need to have some techniques which allows you to see things inside the cell, image them, and make observations. So can we study events inside a living cell? You will say, yeah, of course, use a microscope. So let's see. Answer is yes. And there's some problems. If you start studying things inside a living cell, things get very complex. And when you look at complex things, it's very difficult to make sure that what you observe is reproducible. But the point what I'm trying to say is that what we need to do is find and study a subject in the crowd. This is a cell. And I'm saying in this stadium, which is basically a cell, you would like to study a particular person and to see what he's doing, or a particular set of persons which are coming from the same family, because they are the proteins which do the same function. I want you to study. You say, well, this looks very complicated because I can't even see an individual. So what I did, I zoomed up. I used a much better microscope. And I can say, now in this field, which I'm actually zooming on, there you have your one individual, which is what I want to study. So you said, oh, this problem has not been solved. Why don't we give it the protein a jacket or a tag so that I can track it? So what I'm saying is, let's give the student in this population where I zoom, he's a student from modern school, you give him a purple colored jacket, and we track the purple colored jacket to see what this protein is doing. Correct? And to say, make things simpler, this modern school student in this picture, which is zoomed, I've actually given a fluorescent color. But given that, I cannot put a very big jacket on a protein, I can only light up a small region. I actually painted his nose fluorescent green. So I can track this particular individual using a very nice microscope, which I've got. So now the question is, can you spot this protein which has got the fluorescent green nose? Presumably some of you can, if you stare hard enough, because you're looking at your computer screens, and you can actually peer very, very close to your screen, you might be able to spot a green nose, which is presumably right here, right? But the point is, it's not easy. It's not easy to study the behavior of this protein by spotting the green nose. The problem still stays quite complex. Now I'm going to take a quick diversion. I think I'm running out of time, but I'll try to make it fast. Have you seen this fluorescent jacket? I presume you have. The issue is, these guys which are road workers or construction workers always use this fluorescent jacket so that they're easy to spot. But in the daytime, when you see, it's not easy to spot these guys. Many times you end up hitting these guys with your vehicles because, well, they're expendable. That's what people say. But the problem is the fluorescent jacket is worn to increase spotting, increase their visibility, but it still stays a major problem in the daytime. However, the same problem in the nighttime is not such a major one. So briefly, I'll take a small diversion here to explain the principle of fluorescence, because this jacket is fluorescent. What's a fluorescence? It's a small chemical, a molecule, which absorbs light of a particular energy. Once it absorbs that light, and it becomes, because it's absorbing energy, it becomes unstable, it reaches an excited state. But once it's excited, it becomes unstable. It's like a child who's taken too much sugar. It's very unstable. It's moving very rapidly back and forth. And this motion makes it lose certain amount of energy as vibrational energy. It loses energy. And then it again comes back to ground state. And this process, the light is again emitted out of this molecule. But because it's lost some of its energy, there's a shift in wavelength of light. So what you see here, it absorbed blue light, high energy. It emitted green light, low energy. And the shift in energy, or the light color, is what we call a Stokes shift. And using this property of fluorescence, we can actually control the wavelength of the excitation light and collect very specific wavelength to the emission side, and we can study fluorescence in proteins. So now we have given a fluorescent tag, we've given a fluorescent jacket, we use a higher microscope, the problem is still there. So what we need to do? We need to turn off the light, because if you remember, the fluorescent jackets are very visible in the night time. So jacket solution is darkness is counteracting noise. So even if you've given somebody a jacket, you cannot use just a microscope to study fluorescence, because fluorescence is inherently very weak. There's a lot of ambient light coming. To remove that, we do microscopy for proteins in dark, and you can very nicely see the green nose now. So you can see it's looking very nice. You can actually see very clean yellow nose, and you can see the fluorescence. Now what we have done, we have zoomed up, we have taken the microscope of very high magnification, we cut down the ambient light, and now we ask the question, can I study my protein? And right there, I hope you guys can see a green sharp dot in the same stadium, cellular stadium, but now this time the dot is in the darkness. And now I can study what is happening with this protein inside a living cell. So now what I've shown you, that how we solve the problem by using very smart steps of using high-end microscopes, using fluorescent to track our protein which you want to study, and then turning off the lights and trying to study what this protein is going to do inside a cell. So let's see what this protein can do inside a cell. This is how we tag our proteins. With fluorescent protein, we take our protein of interest, put the jellyfish fluorescent protein, which is green fluorescent protein, either above the protein on the head or on the tail, and we end up getting what we call as a fluorescent protein reporter. And this is the complete spectra of fluorescent protein, which is available to us. Some proteins come from jellyfish, some proteins come from corals, which is fluorescent in red color, and we can use all of them to record behavior of proteins inside living cell. So what do we study? We use fluorescently tagged proteins to study reaction in a cell, pathway, location inside a cell, how protein moves from various parts of the cell, how much protein comes together and go apart. Now, I'm going to quickly take you through this, that this microscope is bad. This microscope is what we need, but the problem is the following. So this is a picture of apple being cut by a person. Our brain is able to fill all the gaps of motion because our brain has seen this process many times over. And this is a very interesting neurological fallacy that our brain does not try to take resolution in consideration when it's looking at things in motion. All of you have phone cameras. All of your phone cameras which are 16, 32 megapixel or even larger. But you're never happy with the quality of pictures it takes. You always say, oh, this is bad quality. But when you look at an HD TV and you are very pleased with the quality of image, even though it is just 1 megapixel, HD is 1 megapixel only. 4K TV is just 4 megapixel, which are like supremely expensive. What is the problem here? What is the analogy here? That when things in motion, which is what you see in TV, our brain uses most of the brain power to fill up the motion, gaps in the motion, so it compromises on the resolution. I'll tell you this simple example. When you take a still picture in daytime, you can take pictures very rapidly, but in the night time, when the light is dim, your pictures are really bad because the motion of light blurs the image. So resolution is inversely proportional to the motion. So if you want to study things in motion, the resolution will be affected. Now, this is a major problem because when you want to study proteins inside a living cell, your brain cannot fill up gaps because brain has no memory, no recollection, no prior exposure to such processes, so it cannot fill up. So what we need to have a microscope camera which can have things with a very high resolution. So if you see here where medical techniques, MRI, PET are very fast techniques. They can show things in motion, very poor in resolution. Microscopy is in a compromise between motion and resolution, but techniques like X-ray crystallography, electron microscopy have zero motion component, but fantastic resolution. So these are still pictures, these are videos, but our proteins are in between them. So that's why we need very high and sophisticated microscope. This is various parts of the microscope, and I'll take you through this. We put cells in which we have introduced our fluorescent protein. We put them into condition in which the cells are happy. We give them nutrition, food, gas through this chamber. We connect it to the liquids and the nutrition and excretory system. We select the light. We choose the light wavelength to extract the fluorescent protein. Pass the light through the microscope. Collect the light which is coming from the fluorescence. Filter out the background noise. Everything done in complete darkness. Regenerate the image, put it to the camera, and we try to image what's happening inside the cell. So what do we see? This is what we see. This is a protein which is on the plasma membrane of a cell. Proteins go from to the plasma membrane from the Golgi, so your protein you can see inside the Golgi also. This is the fluorescent image. That means the protein is fluorescently tagged and is imaged in darkness. So you cannot see anything else but this protein because the only one which is tagged and you can record it. This protein is taken by a camera which can record things at a single photon level, record things at 100 images per second at a single photon resolution, and this can just give you monochrome or grayscale images. The colors are basically indication of how much of the protein is present in that part of the cell. The redder the image, more the protein in that part of the cell. So the plasma membrane is red, this is where the protein is. Golgi, this is where the protein is made and moved to plasma membrane. Now what we're doing, we're trying to see if this protein undergoes changes in location. This protein is involved in sensing chemical cues like dopamine, serotonin. And what we've done here, we've stimulated this cell the acetylcholine. And let's see what happens. I'm looking at this cell from top. I'm looking at this protein, what it does. I put acetylcholine and look what happened. The protein left the plasma membrane. It went to the Golgi. I removed acetylcholine and the protein went back to plasma membrane. A very fascinating movement of protein. This protein we never anticipated to move. But what is somewhat surprising, not only it is moving extremely rapidly. The total time for this movie, 10 millisecond to 100 millisecond. I mean, this protein just moved instantaneously. So this behavior, which we saw by looking at this cell under the microscope, changed the paradigm of signal transduction. So we redefined the paradigm because the time scale in which this event was happening, the protein will always be on a plasma membrane if we actually take pictures or photographs. In 10 milliseconds, if you again take a photograph, it will be on the plasma membrane. But when we start taking video, what you realize, the protein is moving extremely rapidly inside the cell. Plasma membrane, Golgi, back from Golgi to plasma membrane. And a fantastic movie of movement of proteins. So can we see more such movies? Yes. Look at the calcium. This is a cell in which we have loaded a dye, which becomes brighter when it senses calcium. Look at the time span. We had ATP. Calcium comes and boom, it goes away. Time window, less than 10 milliseconds. So calcium is released in the cell in less than 10 milliseconds and reabsorbed. Another video. We flow. This video is disappearing. So, okay, let's skip that. This is a protein which lives in the cytosol, but when stimulated, goes to nucleus to tell cell to change its rate of DNA, the replication or transcription. So here we've stimulated the cell and see what happens. It goes to the nucleus, and when we remove the stimulus, it comes back into the cytosol. Slowly, if you see, it's happening right here. So proteins are very dynamic in behavior. So what else can we use microscopy for? This is an movie of how Mycobacterium, the TB causing bacterium, infects a cell. It was assumed it takes days and months to infect. So in this experiment, the cells were labeled with a green color, bacteria is red, and let's look at this video. Here the bacteria comes and it infects. Total time taken, less than 2 minutes. This is fantastic that you can actually see infection occurring in sub-minute time scale, which is not what we expected to occur in a system unless we looked at it through microscope. So proteins move inside living cell is what you can say. Their movement of proteins is central to their behavior. Transcription factors, signaling proteins, secretory proteins, proteins which move on the plasma membrane, they're all mobile and they're dynamically testing various parts of the cell and trying to take a call that how we can control cell behavior. Not just proteins, molecules, metabolites, small molecules. This is antioxidants like free radicals, reactive oxygen. Everything is mobile, and we can use microscope to study their behavior inside a cell. When you say behavior, their levels, their locations, how they come, how they go away, everything can be studied using microscopy. So ultimately, this is my last slide. Microscopy reveals how proteins regulate cellular function in physiology. It reveals location of molecules inside living cell. So you're trying to study behavior inside a full cell without breaking them apart. It also allows us to study dynamics of movement of protein inside a cell. You can study how proteins are moving, and it also reveals new information about biological pathways, which is otherwise not anticipated. So basically, we are redefining paradigms. So I'll stop here. I think I've overrun my time by almost 10 minutes. I'm sorry about that. But I'm open to questions. Any question in any area which you want to ask, feel free to ask. And for those who slept off halfway through my talk but still have questions and are ashamed to ask now, you can email it to me and I'll be more than happy to get back. Thank you, everyone.
Thank you, Deepak,
Thank you, sir.
for such a very good talk. So if people can briefly unmute and give a round of applause, that would be very nice.
Yes, sir.
Thank you, Deepak, and over to you, Vinayak, to handle the question answer session.
Yes, sir.
All right.
Thank
So
you,
I need
sir.
to figure out how to turn on my video first. I'm struggling with that. I think Vinayak can only turn on my video.
Yeah. Yes, sir. We're doing it. Yeah.
All right. So at least I know not just Ravi is visible, so am I.
Yes. Thank you very much, sir. It's a very exciting lecture. It's brainstorming also. And now we have the question and answer session. Dear students and teachers, now you can ask your questions by unmuting your microphone, or you can comment your questions in the comment box.
Chat box, yeah, that should be fine. Feel free to ask any question. It's not a problem. I understand the talk was a little complicated for the school students,
Yes, sir.
but I'll be more than happy to answer.
Deepak, I will ask a question till that somebody writes there. So in your career, what was the most aha moment you had when you're looking under the microscope? Anything that you can share when you first saw things and were like blown away?
Yes, so the video which I showed you about the protein moving from plasma membrane to the Golgi complex, that was the aha moment. When I showed it to my postdoc mentor, he actually said, this is ridiculous. What you're seeing is garbage. Just ignore it. And we walked out. And when we kept doing it again and again, so one fine day he said, you know what? Since you're able to do it again and again, there must be something happening. Let's start looking at it in more detail. And I think that was one moment because I was trained in microbiology. So looking at a microscope at a protein on a signaling protein was a massive, scale up for me. It was something which I also thought maybe I'm doing something stupid.
No, I
But
mean,
then it was.
At the student level, we don't have access to such microscopes. Although we are microbiologists, we expect to do those kind of things, but we get such opportunities late in life.
Right.
So I think one of the take-home messages is that for students to aspire to come to a level where they can get to access to such good microscopes, so that they can also get their aha moment. I believe.
I think the aha moment, the best thing which I tell with the aha moment, which I like, is be prepared for the unexpected.
You have to believe it.
Yes, and be prepared for the unexpected. You cannot say, nothing is impossible. I mean, that is what anything can happen in biology.
That's right. I agree. And many times what we think, it doesn't happen. So, so I think we have
there are
some
few questions in the chat box.
Please ask.
Vinayak, you can read each one of those out.
Yes, yes, sir. What would happen? Jagdish MG is asking. Okay, what would happen if protein were not existed from Parikrama?
Well, it's an interesting question. It's like more of an evolutionary question. I presume we cannot fathom life without protein because protein turns out to be the building block. But life is an interesting way of finding ways. So I presume if the people say if it's not a DNA life, what would be the case? And people say it will be the RNA life. What if it is not RNA life? It will be the protein life. So I think if protein was not there, somebody else will do the job of protein, and we'll be dealing with a different sort of building blocks. What could they be? I have no guess. I mean, it could be as good a guess as anybody else gets in that. But yes, for sustenance, you need building block, and that's a simple logic. For everything, there's a basic entity, basic unit. For us, it happens to be protein. If it was not protein, it would be something else.
Yeah. Question from Vani. Is there any organisms that cannot be seen under a microscope?
Any organ. So it all depends on what kind of microscope you use. If you cannot see something with a light microscope, you need to have a better microscope. You use electron microscope. But as of now, what is interesting? People have gone to the atomic scale. People have actually been able to see atoms under a microscope. The benzene ring, which Kekule dreamed about, which people study in textbook, has actually been visualized under a microscope by using electron density microscopy. So yes, so right now, I don't think there's anything which we cannot see by microscope because we have reached a level of atomic resolution.
Okay, one question from Sandhya Parikrama from Kormangala. What will happen if our body consists excessive of proteins?
Well, our body is like an engine. It's a metabolic engine. What is excess is removed. What is necessary is retained. So I think that's what the best part of a system, a living being, is ingestion and ejection. So it ejects or basically excretes out what is excess. There's nothing called excess. You can keep eating. The body if you give it more, like the bodybuilders, you give them a lot of protein, it ends up incorporating it.
Okay, and one question from Fatima, again from Parikrama Jainagar. What type of lens are used in microscope?
Well, mostly we use glass. Glass is a fantastic material. Glass lenses are what I use, but it all depends on optical microscopy. So for electron microscope, there are no lenses per se. I mean, basically what you end up doing, you magnify the electronic beam and you collect that beam impression on a plate. So it all depends on what kind of applications which you are looking to. But mostly for simple optical microscopy, it's glass. There's no other material. It's something which is so clear.
Okay. Okay, what type of disease can affect us if our body is refusing to react with a protein from Vani?
Well, I am not getting the question. What do you mean by react to protein? Well, your body is refusing to react to COVID also these days. It's not ready to fight it off, but it's still allowing COVID to cause the infection. So anything can happen. You know, if it's a foreign protein which happens to be a poison, if your body does not neutralize the poison rapidly, then you end up dying. And that's why when you get a poison, you end up getting those anti-sera against the poison which inactivates the protein. So anything can happen. It all depends on how body reacts to it.
Okay, I'll check from the YouTube participants. Is there any
Well, I'll ask
questions?
a question that is, I can read now. Somebody, Sham Parikrama from Koramangala is asking, what is the meaning of phosphorylation and what is its process?
All right. Now, phosphorylation, what do you mean by phosphorylation? See, basically what we're talking about, as I said, if it's a person, an individual, or a person who's wearing a jacket, that is a protein, but the jacket might have logos, and the logos change the identity of that jacket. Like say school, or you're wearing your school jacket. If there's no logo, all jackets look same. But the moment you start wearing a logo in the jacket, it will change, give an identity to that protein. So phosphorylation is like that identity. And that phosphorylation can be added or removed from proteins to change their behavior, because this phosphorylation allows other proteins to identify you differently and change the behavior accordingly. So phosphorylation of the protein is basically like a handshaking mechanism. If you have the tag, two protein can handshake. If you don't have the tag, they will not handshake because they don't recognize you. So it's like a recognition, it's like an identity protein can be given by virtue of these modifications.
I'll ask one more question. I'll just modify it. What Prakasha MB from Government College Chakkur is asking is that how can a small lab incorporate microscope or study proteins? How can they do something like that? Is it possible?
When you say small lab, we all have small labs, but it all depends on the scaling of problems, right? Yeah, so right, it's a very nice, indeed a good question. At a school level, you cannot worry about studying protein. But remember, building blocks means something which you build on. So school is a stepping stone to build a career to look at proteins. So you got to strengthen your building blocks by looking at your fundamentals such that you can study proteins at a later stage in life. So if you want to study proteins right now, it may backfire because you will not have the right foundation which is needed to study proteins. You will not be able to make the right interpretations. So if you're in school or if you're in college, it is very critical that you invest in building your foundations such that you can study things like what we do at a later stage. So every step matters.
Thanks. Vinayak, are there questions in the YouTube?
Yes, from the YouTube, there is a question from Umashri. Sir, do protein in plasma membrane denature?
Every protein can technically denature. When it's a denature, it basically gives an impression anything which is not active is technically denatured. So yes, anything which becomes inactive, it can be considered denatured and it will be removed from the plasma. So cells have got very elegant machinery dedicated to removing redundant inactive proteins from the system. And that's what one of the job lysosome does, to either destroy them so it can use the building blocks, which is amino acid, back again to make more proteins, which is what Ravi studies in various flavors and formats.
Yes. And last question from Abdul. What is the risk of gaining too much of protein?
Well, as I said, the body will not gain too much of protein unless your body is able to incorporate it. But there is something called as a hyperproteinemia, which does happen in people. They basically show signs of excess protein in the system. The liver starts to get stressed, and so does kidney. So it does happen, but our body is very elegant machinery. It tries to get rid of most of the excess material if you are in the prime of your health. When things start going wrong, it goes wrong at many places, and this hyperproteinemia can actually happen in your body.
Yeah. Thank you, sir. It's already time up. Okay. And we came to the end of the session. Now it's time for vote of thanks. On behalf of CNR Hall of Science and Education Technology Unit, we express our sincere thanks to Dr. Deepak Saini for giving us such interesting lecture. And I thank Professor Ravi Manjithaya for convening the program. We thank Professor CNR Rao, FRS, and Dr. Mrs. Indumati Rao for their vision, continued guidance, and commitment to the popularization of science in India. We thank the President of JNCASR, Professor G.U. Kulkarni, for extending his support and facilities for this program. We thank Mr. Dwarkanath for coordinating with the schools and colleges to participate in today's program. We thank all the participants, students, and teachers from various colleges who are present participating in today's event. We thank the supporting staff and the administration of JNCSR for providing the infrastructure facilities and the administrative support for this program. Thank you one and all. Thank you very much, sir. Deepak, sir.
Thank you very much. Bye-bye.
Thank you once again.
Bye-bye,
We'll
sir.
meet in person sometime.
Yes.
Hello, sir. Hello.
Yes,
Please.
So good afternoon, sir.
Good afternoon.
So we'll be getting